Good morning to all. The presentation is Designer Plans for Molecular Farming. Myself, Dr. C. APPUNU, Senior Scientist from ICAR Sugarcane Breeding Institute, Coimbatore, Tamil Nadu, India. The molecular farming, or in bracket the farming in agriculture terms, it means that the use of all organisms, organs, tissues or cells or cell cultures as a bioreactors for production of commercially viable products via recombinant DNA techniques. In simple terms, I am explaining below that the plant molecular farming, the use of all plant organs, tissues or cells or cell cultures as bioreactors for production of commercially viable products through recombinant DNA techniques. This is very simple to understand. The using the plant as a platform for production of commercially viable high value biomolecules in the plant system using recombinant DNA technologies. That is the simple way of telling the plant molecular farming. In this lecture, I am going to cover different subtitles. That includes, one is strategy, the second one is what are the production systems are available, that is the host systems are available, and then the comparative advantages of different host systems and applications, and I take example of some of the case study what we have done it at our laboratory, and then the biosafety issues related to with this plant molecular farming, and finally with conclusion I will stop my presentation. Coming to the introduction, plant molecular farming is the production of pharmaceutically important and commercially viable, that is it may be a non-pharmaceutical products as well, for example diagnostic proteins or industrial enzymes, some cosmetics, etc. These are the products comes under non-pharmaceutical products. Then combination of biotechnology and agriculture to produce new biomolecules for the benefit of human being. It is also known as biofarming or gene farming, because we are transforming a gene to produce its product in the plant system. So it involves, this plant molecular farming involves the plant system, that is agriculture plant, and then the technology, the recombinant DNA technology, through which we are transferring a gene which is responsible for production of high value biomolecules in the plant system. Then the molecular farming started about 30 years ago with the promise to produce therapeutic molecules. This is not a new area of research, but the work was started 30 years back, but still the work is going on to produce high value biomolecules. Otherwise, this technology is highly exploited for production of many non-pharmaceutical products, and some of the therapeutic molecules are very expensive to produce. The question arises, we are producing lot of therapeutic molecules and then that is also available commercially in the market. Why do you want to use the plant as a platform for production of biomolecules? Because some of the therapeutic molecules available in the commercial market, those are which are very expensive for the production, and also when the production cost is more, and then the consumer the retail price also more. In order to bring down the cost or the expensive of the production, this plant molecular farming offers a vital scope. So that also the production of commercially viable biomolecules in plant system comes under the category of green biotechnology. Here I will say the brief history of molecular farming. In 1986, first plant derived recombinant therapeutic protein, that is RHGH in tobacco and sunflower was produced. That was the first product produced in the plant system using the plant as a platform. And then followed by in 1989 the first plant derived recombinant antibody IgG in tobacco was produced. In 1990 the first native human protein produced in plants, that is human serum albumin, in tobacco and potato. In 1992 first plant derived vaccine candidate, that is hepatitis B virus surface antigen in tobacco plant was produced. In 1992, the same year, the first plant derived industrial enzyme, that is alpha amylase in tobacco. During 1995, secretory antibody, that is IgA, produced in tobacco. In 1997, first clinical trial using recombinant bacterial antigen delivered in a tobacco transgenic potato. 1998, first product which is produced using plant as a platform, that is avidin, that was produced in maize, that was commercialized for commercial cultivation, commercial sale, that was commercialized in the market. During 2000, human growth hormone was produced in tobacco chloroplast, targeted to chloroplast system. In 2003, the expression and assembly of a functional antibody in algae was done. In again the same year, 2003, commercialization of bovine trypsin produced in the maize. The commercial level of bovine trypsin was produced in the maize, and it was approved for commercial production and then the commercialization. And then during 2006, the first pharmaceutical commercialization protein, that is plant-made antibody, approved by USDA. This is the first approval from the USDA, that is first pharmaceutical commercialized protein which is produced in the plant system. In 2012, the plant-made vaccine approved for human use. So the vaccine was approved for human use. And then in 2014, a cocktail of 3 monoclonal antibodies for the treatment of Ebola fever was expressed and produced in the plant system. In 2017, they created a small technology, that is plant cell pack technology. That is one of the key advantages of this plant cell pack technology is of microbial and mammalian cell systems is that tests carried out on a small scale generally translate well when the process is scaled up. For example, a high producer CHO cell line in a shake flask generally behaves in a similar manner in a 20,000 liter bioreactor. The link is missing in plant system because there is no direct correlation between small scale test in a cell or tissue culture and what happens to the whole plants in a greenhouse or field due to the much longer growth time, the vastly different environmental conditions in the tissue culture and greenhouse and field scenarios, and aspects of plant growth such as photoperiod dependency which are not present in the cell culture. A new screening platform was recently described which is based on plant cell cultures that are exposed to a vacuum to remove the liquid medium and cast into a porous mass known as plant cell pack. This is the new technology in order to remove the demerits of producing high value molecules in the plant system. Then the subsequently there was BY2 cell lysate protein expression kit was developed. That is cell-free protein synthesis is a powerful method for the high throughput production of recombinant proteins, especially proteins that are difficult to express in living cells due to their toxic or inhibitory effects. The lysates based on tobacco BY2 cells. I have given here to gain more knowledge on this aspect. You can refer the references given below, which is published during 2020 in Biotechnology Advances Journal by Fisher and Buyer. This graph clearly shows that the history of development of the plant molecular farming The plant molecular farming, as previously said, it started from 1998 onwards. First antibody was produced in the plants in 1989, followed by in 1991 first molecular farming SME founded. Then 1998 first clinical trial of a molecular farming product, and then 1999 the first clinical trial of non-transgenic molecular product. Here non-transgenic, you might know about this non-transgenic molecular farming, non-transgenic plants, and this is the first molecular farming product registered as a medical device. And then during 2003 and 2006, this is a peak of expectations based on this plant molecular farming. Bayer buys ICAR Genetics AG to do a lot of molecular farming research, and then during 2007, from that year onwards, the first plant-derived animal vaccine approved by USDA. Then subsequently GMP required for clinical trials, and then in during 2008, insolvencies of several molecular farming. They have started several molecular farms across the globe, and then during 2011 Bayer sells ICAR Genetics AG, and then then during 2011 pharma plant clinical trial started to test the product derived through plant molecular farming, the pharmaceutical products. And then during 2012 there are many agencies, they started to evaluate the product developed through this plant molecular farming, and then the lot of technology has evolved to streamline the increase the production of the product or biomolecules in the plant system. Likewise, there are lot of development in the technology as well as the production, streamlining the production, and then there is a downstreaming process. These are the different aspects involved with this plant molecular farming. As over the period it's reshaped, and then a highly improvised system is available now for production of these biomolecules in using plant as a platform. This is the simplest form of expressing or the giving the idea to you how the plant molecular farming is working in plant system. Suppose example, you can take some of the chemicals or the biomolecules which is of very expensive in the market. You want to bring down the cost of the production, as well as you want to reduce the cost of price of the medicines which is available in the market, otherwise it's expensive. And also in order to this helps to produce large quantities so that this product will reach the unreached people. So this way also gives this social responsibility of this technology. I take an example. Here you see we have the plant system, that is sugarcane. In sugarcane, the sugarcane vacuole is the second largest size of the vacuole, presence of second largest vacuole. The biggest vacuole is present in the celery plant, and then second position the sugarcane occupies. Mainly in sugarcane vacuole, the sugars or sucrose is accumulated in the cell. In this vacuole occupies more than 95% of the cell volume or cell space inside within the cell, and then in this vacuole the sugars are accumulated as in the form of juice. And then this is the primary product. Sugarcane is used for extraction of the sugar. Juice, from the juice sugar crystal is developed. So in addition to this production of sugar in sugarcane vacuole, to increase the benefit to the farmers, we can also additionally produce some of the biomolecules within the same vacuole. So with that idea, we have developed a technology, the vacuolar targeting technology, specific to the sugarcane. and then we have proved the concept that this technology is working well for production of any recombinant protein. You can see here we can target the candidate gene to the vacuole after through genetic engineering approach. Then we have to follow here the two technology, that is biotechnology and then agriculture. Here biotechnology, we make a gene construct and then we mobilize that construct into the agrobacterium or the E. coli, and subsequently first we transform into E. coli, and then from E. coli we mobilize to the agrobacterium tumefaciens, and then we use this agrobacterium for direct transformation of sugarcane by co-cultivating the agrobacterium with the sugarcane tissues, and then we allow them to establish as a plant, and then we test all possible way using the different molecular analysis to confirm the presence of transgene, and also the southern western to confirm the production of the product. This is the one way. In another way, we also use development of transgenic through particle bombardment method. In that method, instead of agrobacterium mediated transfer, we directly bombard the callus with the plasmids with recombinant construct, recombinant DNA construct, and then subsequently we allow the callus to regenerate, and then after regeneration we get the full plant after hardening and all the process. And then again these plants will also be confirmed for the presence of transgene and the marker gene in the plant. Then subsequently we grow them to reasonable height, that is sugarcane is a 12-month crop. We grow them for at least 10 to 12 months. Then from the 10 months to 12 months old plant we extract the juice. You can see here when we go to commercial market we get the juice, cup of juice for 20 rupees, and then this is a pure form of the sugar, accumulation of different sugars like glucose, fructose, sucrose, and the sucrose is occupies a major portion in the juice. And here after transforming making the plant for high value biomolecule production, along with the juice, the gene of your interest product also be available in the juice, and then subsequently this juice is processed using different methods, and then this after dialysis we separate the sugar from the protein, and then the protein is subsequently handled in different way. That will be purified through desalting column, then that will be crystallized using lyophilizer. Then this will be again further purified to the HPLC purification. Then this will be packed into a tablet form or different cubes or whatever the acceptable form in the market. This is a simple way of giving overall idea about the plant molecular farming, how it works, the plant system, how it can be used as a platform for production of high value biomolecules, because the biggest advantage with the plant system is the high accumulation of biomass compared to the microorganisms or the eukaryotic systems like yeast and algae. This is a general strategy to make you to understand clearly see how this technology will be used for production of high value biomolecules. This is the best example. In the plant system, in the plant system there are different tissues in the plant, like you have the root tissues, and then the stem part, and then you have the leaves, and then you have the economic products, that is a cob, example cob, and you have the flower. You see, you have here at least you have 5 different parts or tissues in the plant system. This is an example I'm giving, the maize plant. This this will vary for different crops. Like in sugarcane, you have the mainly the stem is the major part, major economic part, but in maize, the cob is the main economic part. Most of the plant system are suitable for production of high value biomolecules, but the product needs some specific conditions and the specific expression system, so that we can, based on that, we can select the plant or the host. That is, host selection is the primarily major point decides the quantity and quality of the high value biomolecule production. Then the purpose in which you are targeting. Suppose if I want to transfer a protein after making plant to produce, if I want to transfer that protein into a longer distance in normal storage conditions, so we can target that kind of particular recombinant protein to the seeds of any plant. Here I have put the picture of maize crop. You can think of rice, the maize, wheat, or any other crops which produces good seed. So we can transform the plant and then target the expression of that high value recombinant protein to the seeds, and then after harvest of the crop, the seeds will be separated from the cob, or it can be the cob itself can be transferred through general transport facilities like truck and train or buses, and then you can carry wherever you want to supply this product. Or you can carry this product to a particular distance without any specialized storage conditions, and then after you can make a processing, and then you can separate the protein from the seed, or you can directly feed the seed directly. It can be consumed either by human or by animals or by poultry. These are the one example what I have given. These are some of the specific strategies are required for expression of recombinant proteins, because some proteins may be stable in acidic condition, some proteins may be stable in alkali condition, some proteins require always the cool weather condition, some of them may not require cooler temperatures. It depends on the protein nature. We need to decide which part this protein needs to be targeted. So the recombinant proteins can be selectively expressed in particular plant organs such as leaves, roots, seeds, or in cell cultures derived from one of these organs. And then for more detail, you can refer the article mentioned in this slide, that is information published in detail, the annual review of analytical chemistry published during 2016. Proteins can also be targeted to a specific cellular compartment. In my previous slides, I have shown that we can target the protein to a vacuole, and in some of the studies they have targeted protein to the chloroplast, and then some of the study they targeted their proteins to the endoplasmic reticulum, and then some of them have targeted to the apoplast. These are some of the examples where we can target to a tissue specific manner in the plant system. That means that the plant use not only as a platform to produce biomolecules, it has many tissues in it, and then you can choose particular tissues, and then you can target the protein to a particular tissues to protect the protein integrity and then functionality, and then the applications. You can express 2 proteins or 3 proteins together in a same cell, but in different tissues. They're localized in different tissues. When you extract, all three can come together, and then downstream process you can separate all 3 proteins. This is also one of the main advantage for producing high value biomolecules in plant system. That is why the plant molecular farming offers a wider scope to the researchers as well as the commercial farms for production of high value biomolecules. Here you can see in vegetative cells, this is an example for vegetative cells, the protein, the red one circle, can be targeted to the apoplast 1 and then vacuole 2 and then cytosol 3. They can be retained in the endoplasmic reticulum 4. This is it gives some example, at least 4 different tissues we can express. Recombinant protein can also be targeted to the chloroplast by means of signal peptides. How? For transforming for these individual tissues, for transforming the protein to this tissue or gene to these tissues, you need to have a signal peptide. Without signal peptide, we cannot target a protein to a particular tissues on organ. So alternatively, the recombinant gene can be inserted into the chloroplast genome and expressed directly in the plastids. So suppose in case if you don't have any signal peptide which to target into the chloroplast, instead you can directly generate chloroplast transformed events. Directly you can transform the chloroplast so that the production of the protein is restricted within the chloroplast, and then subsequently you can isolate this protein from the chloroplast. So this approach often results in high yields of recombinant protein but does not support certain post-translational modifiers such as glycosylation, because the glycosylation is absent in the chloroplast system. That is the reason this is sometimes some proteins, it is not that all proteins require glycosylation before it is commercialized. It depends on the nature of the protein. Some of the proteins require glycosylation for its functional configuration or to maintain the functionality of the protein. So those protein which requires glycosylation is not suitable for expression in the chloroplast, but non-glycosylation proteins can be expressed in the chloroplast. It depends on that is why I told for specific protein you can target to any specific tissue. Proteins can also be targeted to a specific cellular compartments. Here, when the plants are cultivated in a suspension culture, it is often beneficial to secrete the protein into the apoplast. So through apoplast, because this facilitates purification from the culture medium. That is, you can see it facilitates purification from the culture medium. In some of the cases, digestion of cell wall is an option to efficiently release protein trapped between the plasma membrane and the cell wall. Here you can if some of the cases it depends on many plants have some of the plants have the tight link bond between the cell membrane and the cell wall. Some of them have the loose arrangement, some of them will have them good pore size so that through which the protein comes out. Some of them we need to break the cell wall to release or efficiently to release the protein trapped between the cell membrane and the cell wall, so that we can increase the production yield of the recombinant protein in these conditions. And also here also another example for specific expression. If you take a seed, you see initially we have seen in the previous slides, we have seen there are many subcellular organs. We can target a protein to specific to the subcellular organs. Depends on our requirement and nature of the protein and the nature of the plant system and the availability of the genetic information of the plant. So if we have the target peptides, we can specifically target to a subcellular organs. If you don't have, you can directly make a transformation to transformation. Example, chloroplast transformation you can make, and then you can produce. And then if you take seed as a point or the sink where the protein can be targeted, you need to have a think, then analyze the seed part, which part you want to express. You have here the alluron layer, you have the endosperm, you have the pericarp, you have the scutellum, you have these are the 4 different areas or the tissues. Within the tissue you have the different compartments. You can express the recombinant protein specifically to these different compartments. The seeds are a very attractive site for the accumulation of recombinant protein due to their native storage capabilities. Within the seed, recombinant proteins may be directed to either the endosome or the embryo.